Welcome to the Everything Epigenetics podcast, where we discuss DNA regulation and the insights it can tell you about your health. I'm Hannah Wendt, and I'm the founder of Everything Epigenetics. Today, my guest is Kejun Ying. I had an amazing conversation with him that I think you all are really going to enjoy. We're going to mainly be focusing on causal aging. So rather than correlation of these epigenetic methylation markers to age, what is actually the causation behind it in that process? Albert actually created two new epigenetic clocks called AdaptAge and AdaptAge that we will be discussing. So it's going to talk about your adaptive changes, which are going to be good, and then your changes, which are going to be damage, which of course are going to be not so good and bad in that case. And we'll discuss those applications as well. We'll also be discussing a preprint that Albert just released a couple weeks ago called Clockbase. So this is going to be a comprehensive platform for biological age profiling in human and mouse that is accessible to the public. And a short introduction into my guest today. Kejun Ying obtained his BS degree in biology from Sun Yat-sen University in China in 2019. At that same time, in the same year, he started to pursue his PhD degree in biology at Harvard School of Public Health and Harvard Medical School. He then joined Vadim Gladshev's lab in 2020, and his research now focuses on understanding the cause of aging and developing more machine learning-based aging biomarkers to facilitate the discovery of novel anti-aging interventions. I hope you all enjoy. And now for my guest, Kejun Ying. Welcome to the Everything Epigenetics podcast, Albert. I'm glad you're here today. Thank you so much for being here.
Thank you so much for having me here.
Yeah, I'd love to start. I know I gave our audience just a really brief introduction on you and where you are now. I just want to know more about you, though. So what's your background? How'd you get here? Did you have more of a traditional or non-traditional path in terms of, finding out that you're interested in the subject that we're going to be talking about today?
Okay, so actually my journey to the field of aging is actually quite interesting. So I was throughout my childhood, I was always wanted to be a scientist. Mm-hmm. But at that time, I'm actually thinking about the physicist, so theoretical physicist. But very soon I realized that many physicists, no offense, but they die before their theory is proved to be true or wrong. So I think, okay, this is because it's a hard problem and human lifespan is too short. And at that moment I realized that aging is probably a bigger problem to need to be addressed. And so that's actually the main reason I studied biology during my undergrad in Sun Yat-sen University in China. And I was super lucky. First year in my college, I was able to join a research lab, and they actually study aging-related traits. So they study the telomere, and also throughout my undergraduate, I was actually super lucky to be able to expose to various different subfields of aging. So I was the second year, I was a visiting student in UC Berkeley, and at that time I was in Danica Chen's lab. So they study sirtuin 7. Mm-hmm. And after that, I was in Buck Institute. So Buck Institute is an aging research institute. So I was in Judith Campise's lab. So she studied cellular senescence. And 1 year after that, I went to the UW, so University of Washington. And from there, I joined the Matt Cabering's lab. And I was working on a project related to the rapamycin, acarbose, and brain mitochondrial disease. So this is actually all these experiments I super grateful. So it's allowed me to get exposed to the aging field even before my graduate study, and it's also basically let me know like what I want to study during my PhD. So it's actually boiled down to basically two very basic questions. So the first is that what causes aging, and second is that how do we quantify the aging process? And this is the main reason I joined the English lab. Yeah.
Yeah, well, you have a lot of amazing experience. I didn't realize, I guess, how much experience you had. It's like almost you touched like half of the hallmarks of aging, right?
Yeah.
But because I think that's important. You know, I've always been interested in science since I was a little girl, but it's kind of like it's so broad, you don't know where to start. You really kind of
Yeah,
just have to
exactly.
dive in and get experience at all these different labs, know what you like to learn about and understand. So that's really great. You were able to be exposed to all of that before, you know, your graduate studies now in Vadim's lab at Harvard. So I love what you're studying, and of course that's what we're going to be mainly talking about today is this new paper that you have, and talk about how that relates to epigenetics. So Albert, one of the biggest weaknesses of these, you know, epigenetic clock predictors that are now available is separating the causation versus correlation of course, as you know. So can you speak to this problem? And, you know, maybe before we introduce your paper, introduce our audience to the issue with correlation versus causation.
Yeah. So almost all the predictive model, they are based on the spurious correlation. So basically, they built because your predictor somehow have a correlation with your the trait you want to quantify. So this is actually not a big problem if you're just trying to predict the number itself. So for example, for clocks, if we just want to predict the age of that person, it's totally fine because since the correlation structure is usually stable in the population, your predictor will be reliable for predicting the age. But the problem is that we build the clock not for predicting age, right? Because we know your age. Why do I want to predict that? So what we really want to do is we want to use clock to measure the age acceleration. So basically, we want to say this person looks older than its average. So this also usually can be fine because as long as the person you are measuring are represented in the population of the training data set of your clock. So basically, this person have for example, for methylation clock, this person have the very similar correlation structure of the DNA methylation and age compared to your training data set. But the problem is that when you apply your clock to some unusual intervention, for example, if you have, say, cellular reprogramming, which directly impact your epigenetic landscape, or you have some weird drug or some different intervention that never seen in the training data sets, will be problematic because the correlation structure may be disrupted by this intervention. So I can give a more straightforward example. So there's a quite famous example is that there's a very strong correlation between the chocolate consumption rate in the country and the Nobel Prize laureate number. So this is actually a 2012 Lancet paper. So basically, you can consider this is kind of clock model. So basically, it's you can use chocolate consumption rate to accurately predict the Nobel Prize laureate number. There's no problem because there's an underlying correlation structure. So basically, my assumption is that the economics is related to the chocolate consumption rate, and also separately related to the Nobel Prize laureate. And as long as this correlation structure holds, this model will be accurate. So if you add a new country here, this model will still work, no problem. But the problem begin to manifest is that when you start to do intervention. So for example, one of the country, they say, okay, we want to, based on this model, we want to increase our Nobel Prize winner number. Let's encourage people to eat more chocolate. So, and then based on this model, you will see this country they will have super high predictive value of the Nobel Prize laureate number, but it will probably have no effect because this model use this predictor that's purely based on the correlation. But instead, if we build a model even less predictive, so if we build a model using what we call causal biomarker or causal predictor, so say we use that predictor that really have causal effect on the outcome trait. So for example, for number lowering number, we can use something like a number of university in the country or the quality of the publication from this country. And if we use this predictor, it will be robust against basically any intervention. So for example, if the country say, okay, we encourage people to build more university to increase the quality of the publication, it will really have an effect to your number person lowering number. So the model will stay robust under some different intervention. That's the whole point.
Yeah, I think that the first part of what you were saying there, and you can correct me if I'm wrong, but the first generation clocks, those are trying to predict your chronological age, right? So correlation, causation, maybe a little bit more negligible there. Whereas the second generation clocks that are being created are including an underlying biological aging phenotype, but we need to know if those CpG methylation markers that they're using within those algorithms are purely correlation or causation, and how we start to discover that. So that's really, I think, what you're chipping away at, and is going to be hopefully what we're able to understand more is why is this actually happening? What is aging? I think everyone is trying to understand that question. And I love the example that you gave there. I've heard a lot of, people talk, actually a couple people from Harvard. Dr. Jessica Lasky-Sue is one in particular that sticks out to me, where she gave an example of correlation and causation with, divorce rates and something really silly. And I think that's a good way to put it so people can understand and relate to it more. So, of course, our main focus is going to be just that. You have your paper titled Causal Epigenetic Age, Uncouples, Damage, and Adaptation. So you built the first ever causal aging biomarkers that could separate actual damage and adaptation using the CpGs causal to this lifespan and the health span as we know it. So very, very new subject to the field, and I'm sure to my listeners as well. So, you took this approach by using more of this, what we call this Mendelian randomization. So would you want to introduce our audience to that and maybe how you went about that?
Yeah, sure. So before I get into the Mendelian randomization, let's first start with the causal inference. So what do we mean when we say when exposure or when risk factor is causal to the outcome of the disease or care? So basically, in traditional way, the gold standard to establish causal relationship is using something called a randomized control trial. So basically, it's what all the drug companies doing right now. So basically, you have a group of people. You randomly assign them into two arms, and for one arm you give intervention, and another arm you give placebo or do nothing, and then the effect difference of these two arms on the outcome tree is the causal effect of that intervention. So the key part here, so why this randomized control trial work is the key part is actually the randomization step. So the main reason that observational data are basically say you see a correlation between one phenotype and another phenotype. It doesn't mean that the first phenotype causes second phenotype. It could be just a correlation. So in terms of methylation, for example, smoking can change a lot of the DNA methylation profile in the body, and smoking can also shorten your lifespan. So usually you can see some CpG site associated with the lifespan or with age, but it's actually because smoking have effect on both the predictor or the exposure or the risk factor and the outcome, the lifespan. So for things that have effect on both risk factor and outcome, we call them confounding factor or confounders. So the randomization step actually excludes this confounder. So for example, go back again to this smoking related methylation site. So in a cohort without any trial or without any intervention, you cannot control the methylation value of that specific CpG site. So basically, you will see people who smoke will have higher methylation value, and people who don't smoke have lower methylation value on that CpG site. And you basically have, so here you cannot separate the effect of that methylation site and smoking itself. But if you really want to study the causal effect of that CpG site, you will need to do intervention. So basically, you will need to have some genetic intervention or have some drug to specifically target that CpG site. And the key part is that you need to first randomize your cohort. So you can separate the methylation of that specific site and the smoking. So you can purely look at the effect of that CpG site. But as you may realize, it's not realistic to perform a clinical trial or control trial on DNA methylation because it's just too huge amount of the DNA methylation site across the genome. It's basically impossible to do that. And Mendelian randomization is another approach that takes advantage of the genetic information. So basically, it somehow mimics the randomized control trial. So basically, in Mendelian randomization, instead of look at the DNA methylation itself, we are looking for the genetic variant. So basically, the mutation come from your ancestor that located nearby that specific CpG site. And by meantime, we have another data set that shows that this mutation actually have effect on the methylation level. So you can consider it's kind of like a CRISPR screen, but instead of you doing CRISPR yourself, you allow the nature to introduce different kind of genetic variant and they affect the DNA methylation level at different extent. So you use this genetic variant as an instrument. And why it works is that when those genetic variants pass from parents to the offspring, it's actually they are randomly allocated. So, for example, if one's parent have one genetic variant in their allele, it will pass to their offspring by 50% chance. So this step actually mimicking the randomization step in the randomized control trial. So that's basically how the Mendelian randomization works.
Yeah, so you're taking, let me see if I can get this right. You're taking more of the genetic variant that may be the underlying reason as to why the methylation may be the way it is?
Yes, so we use that as instrument for the methylation itself.
Perfect, yeah, so you're taking all of this very large-scale data and leveraging it. So you're taking the large-scale genetic-based data and you're performing the epigenome-wide Mendelian randomization to identify the methylation sites or those CpG sites for causal age-related traits. Is that a good statement?
Yes, that's correct.
Okay, yeah. Perfect. So you're looking at these CpG sites, and some of these may be deleterious, some of them may be protective changes in aging. So you're trying to categorize these, and why is it important to separate what's causing maybe harm and damage versus what's actually helping? What are we able to know from that?
So this is actually I'll call it probably a myth in the aging field. So people tend to believe that if something changes with your age, this thing is bad. So somehow people kind of believe that. But this is actually never proved to be true or false, but this is not very reasonable because the human body is a very complex system. So when some change happen, so say when some damage happen during aging, your body can there's some program in your gene regulatory network or some genetic program that can act against this change or this damage. So this is something we call response to the damage or the adaptation. So basically, if you look at the change of the say gene expression during aging, there probably a fraction of them would be really doing this damage to your body, and another part of them could be protective. So there's actually was a paper from our collaborator years ago. So basically, they were able to show that the the gene that have differential expression during the disease is actually mostly caused by the disease instead of causing the disease. So basically, all the gene expression change you see during a disease, most of them are our body's response to disease, are not the cause of the disease.
Mm-hmm.
Yeah. So I think and also when you do an intervention, you really want to target those changes that are really damaging your body, but you definitely don't want to target a change that are adaptive. So I can give you a plain example. So like when people get older, they will have basically have a weaker basically vision function. So they will probably wear glasses. So if you just look at the correlation, so people will have a lower, a weaker vision, and people also wear glasses. So there's two age-related change, and you want to have intervention. So okay, let's target easy thing. Let's remove people's glasses. So this is actually an example from Vadim. So I just do it, but I think it's really really good. So basically, when you do intervention, you definitely don't want to first remove the adaptive change. You can remove adaptive change once you cure this damaging change, but you don't want to remove the adaptation first. So that's I think that's a key reason why it's important to understand which one could potentially be damaged and which one could potentially be adaptation.
Yeah. That's a good example. I like that one. I'm going to use that moving forward, and we'll use that clip to explain to viewers. And that scares me. My eyesight, I just went to the eye doctor a couple of weeks ago. I feel like it's already starting to decline, and I would like to think I'm still young chronologically. Yeah. I don't wear glasses or contacts yet. So yes, it's all about looking at, okay, is this actually a response from the disease, or is this the actual cause of the disease? Because, if we're looking at the response of the disease, thinking, or we're looking at the response of a disease thinking it's the cause, then we could be totally leading ourselves in the wrong direction, right? Yeah. So I think your work is super important in this area, and we're going to keep understanding how we can be ready to run, randomized controlled trials for the epigenetic methylation, know what's actually causing these aging factors to move in different directions. So in this paper you actually developed that framework that I've been talking about into these epigenetic clock models, and you constructed, is it just DAMAge? Is it DAM, DAMAge?
Is it DAMAge? Yes.
DAMAge? Okay, I love it. So you constructed DAMAge and then AdaptAge that actually measure these age-related damaging and adaptive changes, respectively. So do you want to explain a little bit about what DAMAge is and what AdaptAge is, and maybe the outcome and what those mean?
Yeah. So, but before that, I want to first clarify that. So during all process, we talk about we call it causal CpG in the preprint, but it's actually so basically it's not proven to be causal. So it's just fit the causal model in the Mendelian randomization analysis. So they're actually putative causal CpG site. So, but it will be like very wordy if you every time you say putative causal CpG site. So we just say causal, but keep in mind it's not really causal, and also for causal site, we are not talking about the site that have direct effect to your aging or lifespan. They go through some pathways. So, for example, some CpG site may increase the rate, for example, the probability of your smoking or your drinking alcohol, something like that. So, some of them may go through the behavioral path, and some of them may go through some more molecular mechanisms. So it's still unclear at this stage. And for the DAMAge and AdaptAge, so this is because, as I described before, we want to separate the potential damage and adaptation. And since we have Mendelian randomization, we can actually estimate the effect of each CpG site on the outcome trait. So basically on the lifespan. And since we have this causal effect and we have direction of causal effect, we can compare this direction to the age-related change direction. So, for example, if you have a CpG site that higher methylation will extend the lifespan, like causal effect. but you see this CpG methylation level actually also increase during aging. This is what we call protective increase or adaptive increase. So this is a good methylation site, and this good methylation will increase during your age. So it's a good. So like glasses. So glasses in general will improve people's vision, and people put on glasses during the age. So it's a good thing. We don't want to stop that. So and the vice versa. If you have a CpG site that decreases the lifespan, and you also see the CpG methylation increase during aging. This is what we define as damaging. So this CpG site is bad and increasing, so we want to target that. And by separating these two groups of CpG sites, we can actually build a clock, two clocks separately. One we call AdaptAge, one we call datamage. The datamage is built purely on using the site we identify as potential damaging site, and vice versa. AdaptAge is we only use the potential adaptation site. So we believe these two clocks can separately capture the damage and adaptation. So one example we found is that we apply our clock to some human cohort and look at their association between the age acceleration based on these two clock and mortality. So we found that the damage actually have a positive correlation with the mortality risk. So basically, if you look older, if you have more age-related damage, and you look older in age, you will have higher mortality risk. But the AdaptAge actually have super weak, even negative association with the mortality. So basically, people if they look older with on this adaptation site, they usually this don't affect their mortality. They will even a little bit negative effect. So basically, they will actually live longer a little bit. So this is I think the main thing. And also we also applied them to the IPS data, so the reprogramming. So it shows that only the damage are reversed during the IPS, but AdaptAge is not reversed; it's even slightly increased. So basically, we believe that these two group of CpG sites they have a very basically different property when you apply them to intervention or to some mortality-related trait.
Sure. So yeah, the AdaptAge, that's going to be the good sites, the good CpGs that we want to look at, whereas the damage is going to be the bad. So we need to separate the two to be able to see how these respond to different things or interventions. And like you just mentioned, one of the interesting findings I thought when I was reading your paper, that the damage is going to be reversed during reprogramming, and the AdaptAge is actually going to be increased. So I like that you mentioned that there. And what do you think the other applications of both of these clocks are? And do they come out as just an age? Is that what you're getting once you're looking at that, or what's the actual outcome that you're receiving?
So the outcome is just age.
Is an age. Okay. Perfect. And what other applications do you think this would fit in? Are these ready, do you think, for clinical interventional trials, or what do you think that these are going to be used for in the future?
Yeah, I think I definitely don't think this clock will replace all other clock. It's just another new clock. But I think what's important is that we emphasize the importance of basically building a causality informed model. Mm-hmm. And we believe that it will be more robust when you're testing new interventions. So basically, if you use a generic clock, so like a regular clock, you see age is reversed, but you actually don't know what that means. So it could be your adaptation is reversed, or it could be your damaging is reversed. So you really want to have an intervention that targeting the damage but not the adaptation. And
yes,
but with adaptation and damage, you will have this at least some ability to separate these two things. And when you're testing your intervention, you really want it to can reverse the damage, but not just the AdaptAge. So we also show that these two clocks are quite sensitive to short-term treatment. So there are some examples, for example, the cigarette condensate treatment or the very short-term some nutrient treatment. The clock is able to show effect at a very early stage. So so basically, I think the ultimate goal for any clock is to use them to discover novel intervention or novel risk factor that could accelerate your biological age. So I think the goal is the same for damage and adaptation. Just you have a better granularity to separate these two very different age-related change.
Yeah, and it makes a lot of sense. I've said this several times on the show before. I spend most of my time digging through the interventional trials that are available and a lot of the research and informing healthcare providers on the data that's available and what looks good. But again, there's not a lot of definitive answers, right? It's a lot of hypotheses still, or kind of we're just learning. It's so new. There's a lot that we can learn from these. But one of my wish that will never come true definitively would be to go back on every single interventional trial that's been created or everything that's been published and run these new algorithms on them that are available. And the data sets are there, but it's just if people are willing to share those data sets, if it's, especially if it's a privately funded study. But I think there's only more that we could learn from that and more insight that we could gain. So I really hope to see these be used in the future and talked about more, just so we can learn more about how they can be used and how they react to certain intervention. Is there any other work then, Albert, that you're doing with the Mendelian randomization at this time?
Yeah, so we're actually also looking at the effect of the so here we look at the DNA methylation, but we also look at basically all the other phenotypes. So not molecular phenotype, just other lifestyle, disease factor, how they affect lifespan, how they affect clock measurement, like based on Mendelian randomization. So yeah, this is almost done actually. Yeah.
Good. You need to send it to me when it's available, and then we'll have to chat later about that.
Yeah.
I'm going to switch subjects completely. I kind of had our little agenda planned out, and then I remember receiving an email from you that you released a preprint a couple of weeks ago that I am so excited to share with my audience. It is called, the preprint is named Clockbase, a comprehensive platform for biological age profiling in human and mouse. Albert, what is this? Tell everyone, what you did, what you created, and we'll go deeper into it as well.
Yeah, it's actually exactly what you mentioned before. So, if you remember before, you said that if there's a many study has been done, and if it would be good, they have data set available. It would be good if you can easily apply the clock on them. So the clock is exactly doing this. So we collected all the data from gene expression omnibus. So it's a huge database, which basically all the researchers upload their experimental data to this platform. So we have thousands of the data set. With each data set contain a very different experimental design. So some people are testing some new drugs, some people testing intervention, and they don't. Most of them don't study aging. Some of them study cancer, study immune disease, but they have methylation data and gene expression data. So since we have the methylation clock and transcriptomic clock, we can actually apply this clock to this data set and look at if there are any intervention that people previously done could potentially decrease or accelerate aging, at least the aging clock. So the Clockbase is a huge database that contains, I think now it has 300,000 sample from Geo. We already calculated the methylation age from based on existing clock, and people will basically, it's a lot of people without bioinformatic background, so even without the knowledge of how to apply clock, they can easily browse the methylation age of each sample just through like the few clicking, and they can also do some statistical tests we have on the platform. So basically, they can compare whether an intervention could accelerate or decelerate the aging process.
Yeah, I think that's the beauty of it. I've been on there and I was playing around with it a little bit, and it's very, very easy to use. So if you're curious, just check it out, and I'll make sure to include everything if you're wanting to play around. I haven't been able to take a deep dive, so I'm setting aside some time to do that this weekend actually. But how did you build it? Did you think, and I guess where did the idea come from? Was it that there is a need and you wanted to make this more publicly available?
So yeah, so this website actually derived from a very small prototype. Before it's called pseudo clock. So it was a tiny hackathon project I did actually with Alex Trapp. It's back in end of 2021, and at that time the website is not as mature as now. So we only at that time it only allow you to upload your own data set, and then it will calculate the the epigenetic age for you. So that's it. We did not connect to any database. But very soon after that, we realized that we actually can do that, right? Because for bioinformatician, I think every time I have some idea, I want to test it, first I look at paper and see, oh, here's a geo data. People done this experiment, we have the data. Let's download the data and test it. And every time you did it, it's actually repetitive. So you just do the same thing to every time you want to study something. And it's become tedious very soon. So I realized, oh, how about I just download all of them, and we just process all of them, and next time I want to use it, I just search them. And after that, I realized, okay, oh, why don't I just publish this? Whoever, maybe there are some domain experts who know this specific intervention better than me, and they can actually look at the intervention they're interested in and to see how they're affecting the epigenetic age. So this is actually the whole purpose of this project.
Yeah, I think it's a great way to build a community and bring people together, right? I'm sure, many people have been reaching out to you already about, different indications that they've been looking at and using your program for. So you identified this issue, you realized you were repeating yourself and kind of pulling up these databases, and then you were like, well, why don't I just make something like this and make it available to the public? So I think it's such a great, useful tool and something that will be able to grow over time. What types of clocks are on there, or what can people expect when they log on? And it's just, is it clockbase.com or what is the link to go there? Is it .org? Clockbase.org. If any of you all are interested, you can check that out and go there. But yeah, when people open it up, what is that journey like? What can they expect?
So basically, I think the first after you click a button, the first page is a UMap plot. So basically, it's a 3D it's a 3D plot where we have each sample embedded in a lower dimensional space. So it's in a 3D plate. So each point is a sample from geo. So we have basically all our hundreds of thousands sample there. And if you put your mouse on each of sample, you can actually see, based on the age methylation clock you selected, you can actually see the age of that sample and what is this sample and what this data sets come from. And then you can, after that, you can, next step would be you select the data set you want to analyze. So basically you can, there's a search box, you can search whatever intervention you are interested in, if it's, for example, smoking or any other fancy stuff. And and then there will just be a table where we pre-computed the clock for each sample, and we also have some metadata in the sample, and you can actually use this information to do some statistical tests on our website. So now we can do some correlation analysis or some t-tests to compare the group. So basically, the output is a quite pretty figure, and you can do a screenshot, and it will be ready for the publication. So it's really a streamlined process.
Yeah, I'm excited to do a deeper dive. So clockbase.org for anyone who's interested, very, very new. And I'm sure it's going to be a great asset that's going to grow over time and just keep building and keep adding. one of the questions I frequently get a lot when it comes to this epigenetic data is going to be about privacy. So I'm sure, I've seen, I think some people ask you about this on Twitter or in other threads. So what would you say to people who really want to use this application of yours, but maybe are a little bit worried about data privacy when looking or using Clockbase?
Oh, we actually do not store the data the user uploaded, so we don't even have this functionality. So don't worry. So the data will just be deleted after I think after your close the session. So yeah.
Good. There you have it. If anyone's worried, they're not even tracking it, so shouldn't be a point of emphasis at all. Well, what is the future for Clockbase? What do you think it can turn into, or do you plan on building on top of that as more information is available?
Yeah. So since we have all this information, all this intervention, and all this sample with their epigenetic age available, the next step would be basically systematic identify the intervention that could potentially, or certain category of intervention that have most potential to reverse or increase the epigenetic age. So, for example, as an example, in the preprint we actually I think one of the biggest problem of the geo data is that the data, the metadata structure is not structured. So people store their meta information in different formats. For example, you have gender column, and someone say, okay, it's a sex, it's gender, and in the column they say female, male, or FM. Someone even put 0 and 1. Nobody know what that means. So I think this is really very it's a barrier, but we are figuring out how to overcome that. And after we can standardize the annotation of all the data, we can actually perform a basically systematic screening of basically all the experiments have been done before, like which experiment can potentially increase or decrease epigenetic age. So one example we found in our preprint is zebulorin. It's a cancer drug. Actually, I don't know. I know nothing about that drug. I just found that in a database, and this drug actually show one of the strongest effect we observe in the whole geo database. One of the strongest.
And what is it called again?
Zebulorin.
Okay, gotcha.
It's a cancer drug, so it's consistently reversing the epigenetic age based on almost all the clock, not almost, basically all the clock we tested, and it reverses by decades. And in the cell line. So we don't know if this effect in human, but it potentially could be something or nothing. But I think it's just demonstrating this potential. So the zebulorin is just one example we happen to found, and for domain expert, they could potentially find a lot of different kind of things in this database.
Yeah. I mean, I just have so many questions when you say, oh, it reversed it by, almost a decade or whatever the exact number is. It's like there can be so many papers probably created off of that one finding and insights. So Clockbase is really hopefully, like you said, a standard annotation people can use, almost like a standard language where people can come together and understand how all of this fits into the epigenetic world and space as it relates to more of those aging clocks. So are you just getting all of this data from public databases, or are people able to submit data to you and you're able to put it into Clockbase?
Both are fine. So currently we only show the data that's publicly available, and people can also upload their data to get the result themselves. We don't save them, but maybe there we can. I'm not sure it's good or not, but maybe in the future we can develop a function that people can choose whether they want to post their data in the database.
Yeah, definitely. Well, I'm excited for Clockbase. I'm just glad we had the chance to talk about it. It's so new, it's so fresh, and I think it'll be a very helpful tool for everyone wanting to know more in this space. And we're getting to the end here, Albert. What are you doing now? You have so many great projects. What are you really trying to focus on in the short term, maybe the next year or so?
So right now I'm at my fourth year. So actually I'm planning to graduate next year. So right now I'm trying to wrapping up everything. And
Yeah.
yeah.
What about after graduation? Any plans yet or thoughts?
I want to be postdoc. So I want to stay in
Okay.
academia. So be scientist. It's
Mhm.
a childhood dream. So I want to postdoc somewhere and keep doing aging research, like from different
Yeah.
angles. So,
Well, I can't wait to follow your work alongside as you grow and follow your dreams. I think that's so motivating. My very last question for you, this is a curveball. You may know if you've listened to any of the episodes before, but Albert, what kind of animal would you be if you could choose anyone in the world and why?
uh, maybe snake.
Oh, a snake. No one said that before. Why a snake?
Because I used to keep a pet snake, so I think looks super perfect as a creature, and they don't need to eat for weeks. So sometimes I
Yeah.
forgot to eat. So yeah,
Well, there's caloric restriction, right? You can just
yeah.
call it. There you go. Well, cool. No one said snake before, so that was super unique. And Albert, we've come to the end of this amazing podcast interview. For people who are listening and want to find you, where are they able to do that?
So I think currently the best play would be Twitter. So I'm
Mm-hmm.
quite active on Twitter. So my Twitter account is just my name, Kejun Ying, Kejun. So it's very easy to find. So yeah.
Perfect. And I'll post that. That way people can stay connected. Well, thanks everyone for listening and joining us at the Everything Epigenetics podcast. Remember, you have control over your epigenetics, so tune in next time to learn more. Thanks so much, Albert. I appreciate your time.
Thank you so much.